start this session on time. As you know, this is the last session, and I'm sure you'll need some time to go check out into your rooms and also get to those flights this afternoon. My name is Sobeida Cruz Monserrate. I'm from The Ohio State University, and it is my pleasure to moderate our session today with my colleague here.
Hi, I'm Colin Weeks. I'm from Massachusetts General Hospital. I'm the medical oncologist there, so we're looking forward to a really great session.
Awesome. So we're going to start. Again, thank you for staying for the last session of the American Pancreas Association. With this session is on multi-omics analysis of PDAC, high resolution and spatial analysis. I please ask that we move the conversations outside. Our first speaker is going to be Dieter Saar, sorry if I mispronounced that, talking about identity mechanism drivers of therapeutic responses in a context-specific mode of multi-omics analysis of PDAC. Thank you very much for accepting the invitation, and I look forward to your talk.
Yeah, thanks a lot for the very kind introduction. I would like to thank the organizers for inviting me to give this talk here on this great meeting. I really enjoyed it a lot. I learned a lot. So it's a great honor being here. My lab is mainly interested in understanding the communication of tumor cells with their environment, of pancreatic cancer cells with their environment. And as Daphne Barsaghi yesterday already alluded very nicely, there are different layers of communication. Of course, first of all, the tumor cells itself, they have different states. KRAS dosage is, for example, an important determinant of cell communication. But there are also, of course, many different cell types within the tumor microenvironment, and we do not really well understand how they communicate with each other and how they promote pancreatic cancer. But then, on the other hand, there's, of course, also the host itself and the macroenvironment, and different features such as the microbiota or host genetics, or toxins, smoking, alcohol, and so on. And they all impact, of course, not only on the tumor cells, but also on the microenvironment and the communication. And finally, we have therapy. And most of the patients receive, of course, therapy, and then they develop response and resistance. And what we are interested in is understanding this whole thing in a holistic way. And we think that this is really important to develop novel therapies. And in my opinion, this has to be a therapy with immunomodulation in order to really be able to cure at a certain time point pancreatic cancer. When we look at tumor cells and their local microenvironment, omics and spatial omics and single-cell omics technologies have been transforming in the last years our understanding of this communication. And that's the topic of the session, and I want to show you some examples how we use these technologies. Last year, there were several really outstanding papers that characterized human pancreatic cancer at a high level. And what we did, and that has been done by Daniele in my lab, so we reanalyzed the data. This is a paper by Tyler Jacks and Avi Fregev. They analyzed 43 human patients that undergone surgical resection. And what we observed is that we get quite a strong overrepresentation in these patients of the classical PDAC subtype. And these more aggressive tumor cell states, such as mesenchymal tumors or neuro-like progenitor states are really very, very rare. And for example, only one patient, for example, had this one here harbored this neural-like progenitor state. So, and that of course limits our understanding of the disease a lot. And therefore, I wanted to come up with the problems we have with pancreatic cancer and and the limitations. And one is clearly the underrepresentation of advanced and aggressive PDAC subtypes or states in our data sets, and also the lack of models for these different subtypes or states that are robust and which we can use in a reproducible fashion. Then we do not have a lot of immunocompetent models, especially not for these subtypes. We do not understand the drivers of these subtypes well. We do not know, is it clonality or plasticity? So what is the contribution of genetics and environmental features? And finally, we do not have really subtype-specific therapies. And that's a bit the space where we're working in. So how can we now disentangle this complexity? I think we need to do kind of a reverse translation approach, generating models for the different subtypes in vitro, in vivo, and also large animal models. And I think we are now there, that we have all that together. What we did in the last 20 years was to generate a lot of cell cultures from mouse models with different genetic backgrounds, different mutation patterns. We characterize them very deeply. And when we look now, and these are now about 900 cell cultures, RNA-seq data, and then we stratified them based on these new 7 subtypes of the Czech's REGEF paper. And there you can see that the mouse space somehow covers the human space, but we have also hybrid states, so that cell cultures share, for example, an Asina-like program and a basaloid program. So we have overlapping states. And I think that's important to notice, especially if we want to model these diseases. And by using now these cell cultures, we then also compared it with the available human data, which we have in the lab. So we integrated PDAC, human PDAC cell cultures. These are about 50, mainly primary patient-derived. And you can see that the mouse space covers the human space, but it's larger, and especially these very advanced stages, so these undifferentiated stages, which are here indicated in blue by this hallmark EMT score, are missing in the human data set, so to say. And that's very nicely also shown here by our mouse model. So we have much more mesenchymal undifferentiated tumors in our mouse model, because these are end-stage tumors. So when the animals are at the human endpoint, and we usually do not have cell cultures from patients at that stage. With these cultures now we can model the tumors and try to understand better tumor-host interactions and communication. And there are, in the end, 4 different layers, I would say, what we are working on. So first, we use these models to understand better the tumor microenvironment, what are the drivers, how immunosuppression is achieved, for example. But we can also do functional genetic screens in different genetic backgrounds, then also in different immunocompetent and immunodeficient backgrounds. We can develop therapies, including immunotherapies, and we can put these mice into different environments or feed these mice, for example, with a high-fat diet, or manipulate the microbiome of these mice. So I think we have quite a nice set now of models where we can really try to understand the mechanisms that drive all these subtypes and that drive therapeutic response and resistance. And I wanted to show you just 2 examples, what we did in the past. So first, better understanding of the tumor microenvironment and the communication. You all know that human PDAC is diverse. We have many different TME states. Nearly every tumor looks a bit different. Some are stroma-rich, others are stroma-poor, others have a lot of immune cells, others have no immune cells at all. What we did is we stratified, or we characterized our mouse cohorts, and we came up with 5 subtypes, so to say. Lymphocyte high, a proliferating, more mesenchymal subtype, granulocyte high, immune excluded, and stroma high. And now we try to use our cell culture resource to model these different subtypes, and that worked quite nicely. So we can now implant these tumor cells into the mouse and thereby really reproducible, generate specific TME states and subtypes. And an example is shown here for the C2 and C3 subtype, single cell RNA-seq data. You can nicely see that they differ in their immune microenvironment really dramatically. So the C2 has a lot of macrophages, whereas the C3 is mainly infiltrated by neutrophils, which have an immunosuppressive features. And this we can also see in the spatial transcriptomics data, but also with more conventional assays like FACS-based, as you can see here on the left side. When we have a closer look in the RNA-seq, in the single-cell RNA-seq data, then we clearly see that these C3 tumors, which are very differentiated tumors, they have a neutrophil infiltration, which is reminiscent of MDSC, so myeloid-derived suppressor cells with a high immunosuppressive score, whereas in the mesenchymal tumors, we see a lot of macrophages, which are have expressed, for example, arginase and have a more M2-like phenotype, which is again immunosuppressive. And I think the striking feature here is that we have very different TME states, very different immune cell infiltrations, but in the end, they all converge on immunosuppression. And that's shown on the left-hand side. So the T cells have a dysfunctional phenotype in all of these different models. By using then the single-cell RNA-seq data together with secretomic data, we computationally analyzed the data and looked for cell-cell communication, and we come up with this scheme, which we are now at the moment experimentally validating. So far, we validated CXCL5 and CXCL1 as well as CSF1 as drivers of these different immune cell states that then recruit the different immune cell populations into the tumor, and then these immunosuppressive immune cells execute then immunosuppression by blocking T cell function. What we can also do with these models is to look at clonality versus plasticity, because we don't know, for example, in tumors that harbor 2 states or 3 states, classically as well as mesenchymal state, we do not know, is it plasticity or is it more clonality? And by using single cell barcoding with express barcodes, we can read out these barcodes. And in the end, the tumors are clones, then also in the spatial transcriptomics data, as you can see here. And there we see clearly that it seems to be more a clonal event, so that subclones have their specific microenvironment, as you can see here for neutrophil infiltration into different clones. So one of these clones is high, the other one is low. Very shortly, therapy development. What we did in the past, we wanted to target immunosuppression and activate the immune system against these tumors. And what Chiara in the lab did, she did a high-throughput drug screen, a combinatorial drug screen, and she came up with a combination of a MEK inhibitor, trametinib, with a multikinase inhibitor, tendanib. Both are in the clinics already. This was highly efficient, specifically in the mesenchymal subtype in human and mouse cell cultures. And when we treated the animals with these combinations, then we observed T cell infiltration into the tumors, which we then thought this might also sensitize to immune checkpoint inhibition. And indeed, in the mesenchymal subtype, and specifically in the mesenchymal subtype, we observed this strong response towards immune checkpoint blockade. And thereby, we were able to target, for the first time, the mesenchymal subtype very efficiently and induced tumor regression. Very shortly, so what is the impact of this combination therapy on the TME? What we observed is that we can induce T cell infiltration into the tumors. That's shown in the lower right part for the mesenchymal subtype, so we get effector T cells in, whereas in the classical subtype, we get the opposite, so there are even less T cells in these tumors. And finally, what triggers this infiltration? We did secretomics and integrated it with single-cell RNA-seq. And what we found is that in the classical subtypes, so we do not see any effect of the therapy, we make this whole thing worse through the combination therapy. So they are high in CXCL2, and by applying the combination therapy, they even upregulate CXCL2 and CSF1, so we get neutrophil infiltration plus macrophage infiltration, whereas in the mesenchymal subtype, we can reprogram the microenvironment towards immune cell infiltration. And CSF1 and TGF-beta are downregulated, as well as CXCL16 is upregulated, which has been shown to recruit TILs into the tumors. With that, I want to finish. Would like to thank all the people that did the work, especially Chiara and Stephanie, and you for your attention. Thanks a lot.
We have time for 1 or 2 questions, please.
Hi, Rosie Sears from OHSU. That was really nice. I'm just wondering what your thoughts are on the mesenchymal data. Looks really, really great, but the fact that the classical, that it maybe makes it worse, and 2/3 of patients are classical. So do you have any insights on how to treat classical tumors?
So I didn't understand the last.
So you were your therapeutic combination worked well in the mesenchymal tumors, but not the classical tumors, right? And I was just wondering if you had any insight into treating classical tumors since they're the predominant form.
Yeah, so we did also drug screens in the classical tumors, and we identified some combinations that were highly efficient. But what we are now working on, I think in the end we need specific therapies for all the different subtypes, or test them at least, and see how the tumor cells react, and also look at how the secretome changes and how the environment changes due to these therapies. So I think in vivo experiments are really important for that. Thanks.
Oh, very nice talk. Thank you so much. I have a question about the mouse model you showed in the mouse model. You have the spectrum for the pancreatitis and then chronic pancreatitis, pan-in, and to cancer. So in terms of the tumor immune suppressive environment, do you, in the single cell sequencing analysis, whether you see in the pan-in situation, whether you see more T cell exhaustion compared to the cancer? So what's the difference you see between the pre-cancer situation or the immunoprofiling compared to cancer situation?
I think that's a super interesting question. We have not really worked on pan-in or pre-cancerous lesion, and if there's a more uniform spectrum of the TME. I think it's a very important point. What we clearly see is that we see that KRAS dosage changes and KRAS amplification occurs very early, and that this is linked to specific environments than in the established tumors. So I would assume that also in the pre-neoplastic state, when these amplifications happen, that then the microenvironment dramatically changes. But we do not have the models in the end, because we have no cell cultures for these pre-neoplastic states. And what we do not know, or what is very difficult, is then to also do the genetics on the single cell level, so to see amplifications of KRAS, for example, copy number alterations, as well as mutation patterns on the single cell level in a spontaneous endogenous tumor. So that's still a challenge, but I think it's a very important question.
I'm going to ask you one question if I figure this mic out. Cytotoxic chemotherapy is used commonly in pancreas cancer, either in the setting of neoadjuvant therapy or adjuvant therapy, metastatic disease. My question is, how do you think or envision that cytotoxic chemotherapy impacts what you're seeing in terms of reorganization of the tumors, mesenchymal versus classical?
Super important question. in the end, there are a few studies that show also that standard of care chemotherapy changes the immune microenvironment and that it induces an anti-tumor, or that it can induce an anti-tumor immune response. But I think there are not enough data so far there. But I think this is really one of the key questions, to look in these neoadjuvantly treated tumors when they are then resected, how and looking before and after how the immune microenvironment changes, and then adding specific immunotherapy or immunomodulators that enhance anti-tumor immunity. I think that's clearly one of the directions to go.
Yeah, I wonder if you can model that with your system, because I think it's in thinking about what you showed, maybe the combination it may be different, right, in the context of chemotherapy versus not.
Yeah, that's what. So what we are currently doing is to use standard of care plus experimental therapies in order to see how we can boost that. Because of course it will be always standard of care will be the backbone. I think there's no doubt about that.
Thank you very much.
Thanks a lot.
So our next speaker is Ashley Keimerman, who will be speaking about 3D reconstruction.
Hi, my name is Ashley Keiman. Thank you to all of you who've made it to the last session. Today I'm going to be talking quickly about how quantitative imaging at the single cell level can actually teach us a lot about Pannin and PDAC development. So we've seen a lot of amazing talks in this past week about how study of CT images and spatial omics technologies can teach us about PDAC and cysts in patients. In my mind, study of anatomy at the diagnostic level, the single cell level, how tissues develop, and as technology has evolved, so too has the field of anatomy. So I don't have to tell anyone in this audience why we study PDAC. It's one of the deadliest malignancies, it's highly invasive, and even in the cases that it's caught at low stage disease, it often comes back quickly, and patients face debilitating pain and distant metastases at diagnosis. So specifically what we develop in my lab is anatomic tissue mapping based techniques to study PDAC quantitatively at the single cell level. And what I'm going to be focusing on today is how we can use quantitative imaging to study Pannin, which is one of the smallest lesions that's known to develop into PDAC. So because Pannin is so small, it can't be seen easily in CT and EUS, so it's typically studied incidentally when it's found in the healthy adjacent tissues of a Whipple. So how do we study the anatomy, the incidence, and the omics qualities of Pannin? We had to develop a new technology. So at Johns Hopkins, we've been developing this technology CODA, which uses deeply sectioned histological slides to study tissues quantitatively. And I'm just going to quickly bounce down the methods of CODA and then show some examples of how we've used it to study Pannin. So I don't think I have to tell this audience what H&E is, but I love this animation, so I'm going to show it. Histological sectioning allows us to take any tissue, cut it into thin two-dimensional slices, and the foundation of diagnostic pathology is H&E staining. So H&E is a pH-based stain that is somewhat agnostic and lets us see a lot of things at the single cell level. So hematoxylin is going to stain the acidic components of tissues, so that's going to get you all the nuclei that contain DNA. In pink, the eosin is going to stain the basic components, so the collagen, the smooth muscle, the cytoplasm of cells, and all the enzymes within the cytoplasm will pick up a bit of both, giving you these beautiful violets. So this mix of all these stains lets you identify a lot of components in H&E. A pathologist can look at this slide and tell you that this is a duct. They can tell you that the top of this duct is neoplastic, this is a Pannin, and the bottom is lined with non-neoplastic epithelial cells. So looking at H&E, a pathologist can see a lot of things. What we want to do with CODA is to quantify them. So with CODA, we'll deeply section a block, stain a subset of the slides with H&E, and then apply a set of computational techniques to build these back up into a digital volume. So step one is just taking, say, 1,000 H&E slides and quantitatively aligning them into a volume. Step two is what I call quantifying the pathologist. So can we use AI to map quantitatively the things that a pathologist could identify in this image? So can we label the pannin, the ducts, the acini, and so on? With a combination of all these things, the next question was, can we study pannin? So using CODA, we can go down a block of tissue and exhaustively outline all of the pannin in these images. You can see how beautiful pannin is, how complex its connectivity is. Zooming out, we can see what does this look like. So pannin is this complex octopus that exists in the ductal system and can squeeze out of the ducts into the adjacent lobules to look somewhat hollow and tube-like in some locations and somewhat solid and clumpy on the edges. So next question, if we can map pannin in these small data sets, can we now map it in big data sets? So thousands of slides, whole slide image level. Yes, we can. In this video, you see zooming down a stack of 1,000 H&E slides, automatic identification of, in this case, 55 independent pannin lesions. In a second, the H&E will fade away, and you'll see what all these look like. So we have all of the pannin manually identified within the non-disease ductal system. So we applied this to one sample. Next, we applied it to almost 50 samples. So in the healthy adjacent tissues of Whipple's, so in patients diagnosed with things like PDAC, PNET, and so on, can we look at the adjacent tissues to see these cysts that develop? In each of these images, in blue is outlined the non-diseased ductal epithelium, and anything that's not deep blue is a pannin. So I can highlight a sample that contains quite few pannin. Some individuals had quite few, but all of them had some amount of pannin. And some samples just had tons and tons of neoplastic content. So unexpectedly huge amounts of pannin in these blocks. So I want to walk down a couple of findings in the anatomical mapping of these blocks and then jump into some genomics technologies that we've applied as well. So conclusion one is just that pannin exists in shocking abundance in human tissues. So on the x-axis is each of the individual cases that we mapped, and each dot is a pannin that we found in one sample. This is to show that many of the cases contained pannin with well beyond 500,000 cells. So pannin can get huge, and yet when we study them in 2D, we can miss this fact. Second, if we look at the amount of the ductal system that's composed of pannin cells, again, the mean of these samples is that well beyond half of the ductal system is pannin. So when we think of pannin lesions, we know, well, we believe that most PDACs develop out of pannin. We also need to acknowledge just how common pannin is in human tissues. So because we modeled blocks of human tissue, not the whole pancreas, we attempted to use this data to estimate how many pannin individuals could have, and we came up with a mean estimate using this data set of 1,000 pannin. So again, just emphasizing how extensive, how common, how many pannin we all have. Taking a step into a side study, so because CODA can map these tissues so agnostically, we had a lot of additional findings as well. So because we modeled the healthy adjacent blocks to Whipple's, many of these patients had diagnoses including PDAC, though the tissue that we analyzed had no PDAC on the edges. So if we found incidental PDAC cells in the middle, it was unexpected. Because we mapped these blocks at the single cell level, we unexpectedly found in one of these samples just the tiniest little pellet, less than 1,000 cells out of a block containing 1 billion cells, a small venous invasion. And while this might seem somewhat incidental, what it's evidence of is why in so many PDAC patients who have a Whipple done, why does it come back locally so often within 6 months? It's because of, in some cases, things like this. So it's not humans making mistakes, leaving huge swaths of tissue behind. It can, in some cases, be these tiny little seeds that escape the main mass and can go colonize and build new PDACs. And again, just to emphasize, this was such a small amount of tissue, no pathologist is going to find it. It existed in 5 slides out of 1,000. It contained less than 1,000 cells out of 1 billion. So this is just tiny little seeds that these lesions leave. Finally, I'll move a bit quickly. So using the unstained slides with CODA, we can also label immune components. So with this, we show that Pannin lesions not only exist in huge incidence in human tissues, but have quite complex inflammation. So while we think of PDAC as a cold disease, we show with Pannin that an individual lesion, this big octopus is one Pannin, we can have cold locations that have almost no inflammation. We find that a lot of these tend to be in the dilated locations of the ducts. We don't quite know what to make of that yet. And in adjacent locations, you can have a hot location of Pannin that has 5,000 times as many immune cells. Some of these seem to be occlusions of the duct, locations of ADM, but again, just to emphasize, Pannin, inflammation, PDAC is not as simple as we might think it is when we study it in 2 dimensions. The last finding I want to show with the inflammation is that if we look at the individual Pannin level, we see no link between each of these dots as one Pannin, between an individual Pannin's volume and the amount of local inflammation. But if we zoom out and we look on a global scale, each of these dots now is one human sample. The x-axis is how much Pannin does that sample have, what's the global Pannin content. This links highly to global inflammation. So inflammation in Pannin is not something local, it's something global. And this links back to the cycle of inflammation causing Pannin, Pannin causing ADM, causing inflammation, and so on. Finally, in the last 2 minutes, I want to talk about how, because we have all these unstained slides with CODA, we can link not just to IHC, but to genomics as well. So by mapping individual Pannin lesions in the H&E, we can identify back onto the 2D slides, this duct is Pannin A, this duct is Pannin B, and so on. By combining this with LCM, we can ablate distinct lesions and distinct locations of each Pannin into distinct vials. So this allows us to look at the somatic mutations of spatially independent Pannin in a single block, and to look at subpopulations of clones within the same Pannins. So I'll quickly walk down these findings. Initially, what we found, say in a sample containing Pannin A, B, C, and D, and an adjacent PDAC in a lymph node, to explain this table to you, each of these Pannins on the top matches to one of the lesions you see in the video. On the left-hand side, we have listed the somatic mutations we found any place in any of these samples. This column is showing you that in Pannin A, we found a G12C mutation in 2 of the 5 zones. So the conclusion of this slide is to show that spatially independent Pannin tend to contain spatially independent somatic mutations. So these Pannins developed independently. In fact, most Pannins we mapped tended to have independent mutations. Second, while most Pannin had independent mutations, we did find cases of spatially adjacent Pannin containing the same mutation. So I also want to note that this mutation, Q61H, is highly uncommon in Pannin, so it's unlikely that this was a coincidence that these all developed independently and just happened to be close by. So we take this as evidence that some Pannin can seed off individual cells that will move slightly down the ductal system and become a second lesion. Finally, when we looked at the biggest Pannin in these samples, we found many cases of the same Pannin containing multiple mutations. So the hypothesis we have is that because Pannin exist in this enclosed ductal system, multiple Pannin can develop and fuse to become these giant lesions. So in conclusion, we've shown that CODA can map the incidence, the anatomy of human tissues at single cell level, and it's highly amenable to study of things like Pannin, which can't be seen in diagnostic imaging and can't be labeled specifically using antibodies. By combining with genomics, we show that many Pannin develop independently, but that they can fuse into these massive lesions that we find. And lastly, I want to thank all of you who came to listen to my talk, to the team at Johns Hopkins, and to all the people listed who've helped with these studies. Thank you.
Do you have some time for some questions?
Yeah, it's Dr. Wang from MD Anderson. Very nice study. I have 2 questions for you. Did you compare the Pannin lesions from the ductal carcinoma samples versus those from endocrine tumors?
We did. About half of the blocks we analyzed, the patient had a diagnosis of PDAC, and about half of them had something else. I think about 5 had a PNET. We found a slight uptick in the Panin incidence in patients with PDAC, but it wasn't as big as we expected it would be. We actually still found quite a bit of Panin in individuals with PNETs.
Thank you. The second question I have, you show this nice, modeling for the satellite tumor foci in the block. Did you do any kind of LCM molecular study, compare, the satellite tumors? Are they different or same with the main tumors?
Yeah, so 2 of the samples we analyzed also had PDAC in the block. In one of them, the PDAC was physically touching the Panin, so we assumed they would have the same mutations, and in that case, they didn't. The second instance, we had a Panin lesion and an adjacent PDAC within a lymph node, and in that case, they did have 1 mutation in common.
Thank you.
Mhm.
Ashley, excellent work as always. One of the things that I was kind of struck by was the large Panins. And one of the issues that we always deal with in the pathology level is it a Panin, a large Panin, or is it an IPMN? Especially when you deal with polyclonal or polyclonal KRAS alterations, are you really seeing an IPMN? Can you distinguish between the two using CODA?
That's a good question. So all the blocks we analyzed we specifically avoided cases that had a diagnosis of IPMN, but I actually have a couple slides that I cut today that in the 50 samples we analyzed, in 20 of them, we can find something that pathologically is called a Panin, but that we can simulate a histological cut that would make it into an IPMN. And then we went back to Elliot Fishman at Johns Hopkins, and he looked at the diagnostic CT. And in those 20 giant Panin, as we've been calling them, he can see half of them in CT. Okay. If he looks specifically and long enough, he's seeing something distinct of the PDAC that looks like it's in the ducts and could be these uncommon huge Panin. So I think some of how we think of Panin as small, as uncommon, as not seen in diagnostic imaging, maybe isn't the case.
Can I ask one last question? Now that we're in the neoadjuvant era, and I love the pictures, but they're not treated pancreatic cancers.
Yeah.
Can CODA deal with treatment response in determining like where the edges of the pancreatic cancer are, especially with the associated fibrosis? Have you tried neoadjuvant treated pancreatic cancers at all?
Yeah, we have ongoing studies actually looking at exactly that of can we look in neoadjuvant blocks of PDAC and spatially identify using the H&E the most affected and the least affected locations, and then see using omics techniques what's going on. Like why did we fail in the clinic almost?
Very cool.
Thank you.
Yes, sir.
Yeah, Michael Zendelman from Weill Cornell. A history lesson and a question. So in the 1980s, when we started doing real pancreatic resections, there was a subset of surgeons who thought that pancreatic cancer was multicentric. So just be careful that you may be resurrecting a really bad thing. And there were randomized controlled, not randomized trials, but trials showed that the total pancreatectomies had no benefit in survival than the standard Whipple. But it was a really big deal back then. The question is based on what you're seeing and the paper that was presented yesterday from your group about the creeping of the tumors in the ducts, are you changing the way you look at specimens from the operating room? Thanks.
That's a good question. I'm not a physician myself, so I can't speak to how it's changing what we see in the clinic, but I hope that this could have an impact in how we assess PDACs clinically.
Real quick question, Jose Trevino from VCU. I mean, the work is pretty shocking, right? I mean, at the end of the day, as a surgeon who cuts this out, I mean, my margin being negative might not mean much at the end of the day. Yeah. So how does your group at Hopkins translate this? This is great stuff. I mean, this is really telling us a lot about the biology and how these early lesions could have genetic changes that can make a difference within what should be a quote unquote normal pancreas with a negative margin. So how is your group even anticipating that? And, you know, for future, I mean, is this going to change our practice? And I get the prior comment about this really being a systemic disease and might not matter so much what's happening in the remainder of the gland or local recurrence, because systemic is what kills our patients. But is your group doing anything about that? And again, wonderful work.
Yeah, so we've been thinking a bit lately about, since we've shown how extensive Panin can be in human tissues, can we link it somehow to diagnostic imaging? So do the giant Panin, can they be seen in CT? Can even if you don't have these big lesions, can something with AI, with all of this extensive quantitative mapping we can do in histology, can that somehow be linked to something that maybe isn't a visible cyst, but something that's diagnostic? And that is the way that I hope this is going. But it, yeah.
Quick one. Thank you. Are you doing fantastic work, by the way?
Thank you.
Trying to map the sub-classifications of Pannin? Because I didn't hear much about that from you yet.
Yeah, so we have a couple ongoing studies in that. So we have some looking at the 2 grades of dysplasia. Can we map those anatomically, how they connect with CODA? We've also looked into what we call phenotypes of Pannin. So based on its location in the ductal system, in locations of ADM, in locations of dilated ducts, we find that these don't have the same mucin content. They may not have the same immune components. So that is something that we've been seeing if we can spatially map the complexities of individual lesions.
Thank you.
Thank you.
So just one more quick question. So I'm a naive medical oncologist, but my question is there any relationship between the Pannins, the RAS mutations you're seeing in the Pannins and then the primary tumors? And does that have any impact on those patients who may develop metastases or recurrent disease?
In some cases, we found that. In the 7 blocks that we did the genomics in, 2 of them had a PDAC in the same mass. 1 of those seemed to be connected to a Pannin lesion and 1 of them didn't. I think mainly what this shows that's so shocking is that so many Pannin do not develop into PDAC. So if we can identify what's unique about the ones that do, maybe we can see which Pannin is it okay to have and which Pannin is it not as okay to have.
Very nice.
Yeah.
Thank you.
So much.
Thank you. And now to our last talk of the meeting, Dr. Arsen Osipod from Cedars-Sinai.
Thank you. Thank you to the organizing committee for inviting me. I know I'm standing between you and enjoying the rest of San Diego after this amazing conference. So I'm going to be talking to you about a multi-year, multi-institutional project, which is entitled Multi-Omic Profiling of Pancreatic Cancer Integrated with AI for Clinical Outcome Prediction. I don't have any disclosure, so we'll get right into it. So, there are challenges that span all of cancer, but are really highlighted by pancreatic cancer. And we know these challenges because in pancreatic cancer, there's a lack of effective predisposition markers. These are who should we watch for the development of the cancer. There's a lack of predictive markers. This is, what to treat patients with once they have pancreatic cancer, prognostic ones. We don't have great ones. We have typically CA19-9, which we're all aware of. And this really was invented in 1978 or discovered. And this is what we still use in clinical practice today. And then ultimately, are there markers that can help guide specific targeted treatments? And this has changed with NGS. But we talk about precision medicine and all, but what does this really mean? And nowadays, when we think of predisposition markers, some of the actual precision markers are germline genetics, which we routinely now do, environmental history. There are prognostic markers, which are really based on path that we look at to see if there's lymphovascular invasion, perineural invasion, number of lymph nodes, et cetera. We have predictive markers, again, that sometimes can be based off germline genetics or somatic mutations, CA19-9. There are newer markers that are in development that we've spoken about at this conference, which include circulating tumor DNA, lipids, proteins. These are research questions that still stand. But as of today, and generally in the community, minus the great research that's been presented here, our approach to precision medicine is what we really describe as unidimensional. We take maybe tumor tissue, some clinical information, and genomic information, and we try to use that to predict how patients will respond or do, or what their prognosis may be. But I think our goal is really to shift forward and to do what we describe as multidimensional analysis. This is where, and we use this example, you have a 3-dimensional shape. Don't look at one surface. DNA and clinical information is really one surface, is to look at it in 3 dimensions, multiple sources of molecular information to help guide us for predisposition markers, prognostic markers, and predictive markers. So with this challenge in mind, we developed something called the Cedars-Sinai Molecular Twin Platform, which is what I'm going to present to you. So how did the platform come to development? So we have a patient, and the patients today go through our pancreas cancer multidisciplinary clinic. And these patients, once they come into the clinic, we have curated notes that we capture their clinical information in great detail. And then simultaneously, we obtain a tissue sample and serum samples that we ultimately will want to expose these to deep molecular profiling, and I'll go into details of what that entails, as well as to computational pathology. Then we take all of this molecular information and we apply a very unique AI-based algorithm, and we use external databases to validate the findings. And there's a simple goal, is to see if we can create a computational and molecular replica of the patient to see maybe there's a patient that historically we've treated that has a very similar molecular AI-guided profile that can help us treat the patient today, with the goal of predicting, prognosticating, finding novel targets that are very patient-tailored, or helping us guide for trial selection. So we actually completed a proof of concept study in pancreatic cancer that's shortly going to be published in Nature Cancer. And what we did is we started with 74 patients with stage 1 and 2 resected PDAC. And we took, applying our molecular twin platform, we analyzed their NGS, we did whole transcriptome RNA-seq, we did tissue and plasma proteomics, as well as lipidomics, computational pathology, and their clinical data. And we analyzed this all together. That led us to 6,363 individual molecular and clinical features. Then we combined this into a very unique multi-omic model with 3 very specific goals. Can we use it to predict disease survival? Can we find clustering of patients that may molecularly match each other? And can it begin to give us insight about specific pathways that may be relevant for prognostication or for targeting? And the way we did this is, when you take all 6,000 features, you actually have really 10 analyte sources. When you do the various combinations, that leads you to about 10,000 combinations. And we had 7 different machine learning models, which included deep neural networks, logistic regression using PCA, as well as a recursive feature elimination random forest, just to mention a few. When you apply these 7 to the 1,000 combinations of analytes, that leads you to over 7,000 what are called grid research runs. This is the computer algorithm running every potential combination of all these molecular analytes. And then using a leave one patient out approach, we trained our data set, and then we validated it on 4 separate cohorts from TCGA, Massachusetts General Hospital, as well as 2 separate cohorts from JHU with our collaborators. And I'll give you an example of starting with the single omic sources. So computational pathology is one very unique aspect of our approach. So we take the digital pathology slide, we scan it, and using an AI algorithm, we look at nuclear architecture, size, shape, hematoxylin intensity, distribution, nuclear proximity, as just one example of a single digital source for pathology. And then we combine these features, apply our algorithm, and just using one single omic source, it tells us that we can obtain an accuracy and positive predictive value of 0.66 and 0.76, which is okay, at least for using digital pathology to predict a disease outcome. But that's just one example. We said, what about if you combined all the single omic sources? So this table is actually a very good representation, and I'm going to start from the bottom up. So we took CA19-9. So CA19-9, we routinely use it, and we said, if you take a CA19-9 preoperatively, how well does that predict disease outcome? And the accuracy and positive predictive value was around 0.6, not that great, and that's why it's at the lowest level there. But as you go up higher, you'll see each specific single omic source performs better using AI rather than CA19-9 to predict how a patient will do. The top single omic predictor was actually plasma proteins. And the best predictor was when you combine the entire multi-omic data set of DNA, RNA, protein, lipid, and digital pathology with clinical information. This, again, is a very potent technique, and you can see that even with plasma proteins, the positive predictive value of it and its accuracy is closer to 0.8, much better than what we would achieve with CA19-9. Now, then we took a different step. We said, well, do you really need to have all this molecular data? Does it help to have more or less? And if you look at the top violin plots, you can see that the middle section actually has the highest number of accuracy, which tells us one thing. If you add 9 or 10 more analytes, it doesn't necessarily improve your model. If you have 1 or 2, it's not enough to predict how patients will do. And then we said, well, which ones are important? So the graph at the bottom, which has multiple colors, and I want to highlight the green color, which actually represents plasma protein. These are the top 15 multi-omic models for disease outcome prediction. And it shows that there's a lot of complementarity to it. All of these models have relatively close accuracy and positive predictive value, but they have various combinations of analytes. Some are more driven by protein, some more driven by RNA, but we can see the biggest driver across all top 15 multi-omic models is actually plasma protein, which was our single omic source that had the best disease outcome prediction. And this is really important because it tells us that really you don't necessarily need to do a 6,000 feature molecular analysis. And this has big implications because this type of analysis, there are a handful of institutions globally that have both the resources and they can go beyond the cost constraints of doing such in-depth molecular analysis. So that posed the second challenge. And we said, can we use AI to develop a model where you don't necessarily need to have all 6,000 molecular features to predict disease outcome? So we developed something called the parsimonious model. So what is this model? And I'll walk you guys through this. If you look at the curve, you'll see on essentially the right side, you're starting with 6,000 features, all 6,000 molecular pieces of information: RNA, DNA, protein, lipid, clinical data, computational path. And we used a recursive feature elimination strategy where we went one by one, removed each single feature and ran the algorithm again. And we kept doing this. And there's actually an optimal point where you can still achieve a very similar accuracy and positive predictive value to the full multi-omic set, but you only need 598 features. So one, you save money, you save time, and this is something that can be applied potentially in resource-poor countries or resource-poor locations. Has immense implications. And again, this is really probably the most important part of our finding, that AI and molecular profiling can come to a point where you can possibly take one digital pathology slide and a sample of blood, analyze a handful of features, and still be able to predict disease outcome as an endpoint potentially. So the other part that we found that is important, we use pancreas cancer because this is an institutional interest of ours and an area of excellence for our institution, but this platform really can be applied to any tumor. It is really tumor agnostic. And it shows that as an endpoint for disease survival, multi-omics has a potential for prediction that's better than our current standard, which is CA19-9. And I didn't get a chance to go into this, but we can actually cluster patients to see their molecular profiles, how patients match to other patients historically to give us guidance about which treatments may be better for them. We found even certain pathways when we did gene ontology network analysis that are relevant, such as CXCR4, mTOR, that actually came out through the paper. And this really has implications for trial selection and for drug discovery. But we also realized in doing this work that there are some limitations which led us to how we're going to approach this in the future. So we use 10 analytes from DNA, RNA, protein, et cetera. And we've talked about this even at this meeting about how important the tumor microenvironment is. We didn't explore it in this particular iteration, but in the future iterations of the molecular twin, we plan to incorporate in-depth tumor microenvironment analysis as a single omic source. Dr. Fishman yesterday had a great talk about radiomics, and it actually raised the question of us working together to use their AI for radiomics as another single omic source. You can incorporate wearables and ctDNA. Another kind of a shortcoming of our study was we took one data point from one time, which was a pre-surgical sample and the surgical specimen. But our future approach is actually to do multiple time points throughout a patient's treatment course, incorporate that into the AI algorithm, and that will lead to better performance. And again, in this pilot project, we used disease survival as an outcome, but you can choose other outcomes: disease-free survival, progression-free survival, response to specific chemotherapeutic agents. So with that, I'll conclude. I want to think this is a huge team effort, and we have great collaborators at MGH and at Johns Hopkins that helped with that, and my mentors, Dr. Pandal and Dr. Zhang. Thank you.
Open for discussion, questions? Well, I guess I can get started. When you mention protein and metabolomics added into the system or being incorporated into the system, what do you mean? What platforms are you talking about? I mean, from a basic science point of view, you can use like metabolomics. You can have 10 metabolomics investigators in the room, and they're all gonna disagree on a platform. So how can you standardize that, and what are we talking about there?
Yeah, that's a really good question. So, you know, I focus on the tumor microenvironment. So there's many ways to analyze the tumor microenvironment. For example, you can use simple IHC, you can do multiplex IHC, you can use other technologies that they use with like IMC, for example, or even single cell analysis. The versatility of this platform allows you to actually add any particular technology or single omic source and test it. So you can take the data, test it, and see, does the data actually work? Can you validate it? If it doesn't, then it drops in its kind of importance within that multi-omic modeling. So we leverage AI, and in fact, we've tested, there were some other analytes that don't really hold up. So the AI tells us likely what's going to be more important. So if there's a question among investigators of which one has more clinical importance, this would be actually a good platform to test that.
Great.
Hi, this is Anu from Mayo Clinic. Thank you for the outstanding presentation. I really appreciate the work you're doing. So you mentioned there are many data sources you're using to build the models and extract the features. So you shared that you use EPIC data, so clinical data. Could you share a little bit more about what parts of EPIC or what kind of data?
Yeah, no, this is a great question. So actually what we've done at Cedars, we've templatized, and we did it as some pancreas cancer. So actually I started this at Johns Hopkins when I was a fellow there. The clinical notes are templatized. You don't really type a lot in. It's a selection menu about staging, about their path, et cetera. So there's no error that happens in that. So we took 25 very relevant information that's already in EPIC, including stage, the path information, their family history, which is already pre-populated in there. And that was the information that came from EPIC, as well as secondary diagnosis that patients had that was already within EPIC.
I see. So another question, one of the ways I was thinking this could really help is early detection. So if you look at the patients in a case control study and look, you know, a little early, like a couple of years early, and see how that data kind of looks for them, and build an AI model to see which features are kind of you know, has a more influence on somebody getting cancer versus not. That would be a great study. But again, I know this is a lot of work, and I appreciate what you're doing. I just thought it'll be a good idea.
It's actually a great point. This has even come up internally. You know, we have a high-risk pancreas cancer group and Dr. Kaiman presented great work about PANNs, for example. Can you take that type of omic information and incorporate it with your outcome being the development of PDAC? So there's a huge potential, but as you can see, it's extremely resource intensive.
Yeah. So, right. Well, thank you so much.
Well, thanks everyone. I'm going to turn it over to our president, Dr. Lee.
Great talk, Arsen. What a section to end the day and end the annual meeting. Thank you, Zubeda and Colin, for the outstanding work. Moderating this exciting section, we really save the best for the last. And I want to thank everyone who stayed until the last minute. Look at the room. Last day of the 3.5 day meeting, we still have roughly 100 people left. This shows the passion and engagement from our audience, from our community. This shows the love to APA. This morning we ordered enough breakfast for 150 people. We end up having to order 50 more. So that's how much people want to come and stay until the last minute. I really appreciate that. And there's never a dinner that will never end. So it's time to say goodbye. And I want to thank all the speakers and the moderators for the outstanding talk and for keeping these all the sections run as smoothly as you can get. And literally, this year we cover many new topics, very special and inspiring topics, especially on cancer, cachexia, and neural crosstalk, and AI in pancreatic cancer. And I've received a lot of feedback, very positive feedback. The program was really special. I'm thanking all the committee for doing that. Again, I hope you all enjoyed your stay in San Diego. I want to invite Dr. Saluja to come to the stage, give you a quick update and welcome for the next year's meeting. Dr. Saluja.
Well, I also want to add my thanks to the brave souls. After 3.5 days of almost 24-hour things here, I mean, Thursday we were here till 11:30, 12, and then Wednesday we were here late, and last night was again. So really appreciate that. And we had a great meeting. I mean, really, science was great, ambience is great. But what I'm going to tell you is that next year it will be, if there is anything better, it will be better. I'm not sure how we can make better than this, but we will. At least, it's Maui, Hyatt Regency. I've gone there a couple times. It's a beautiful property. All of you will enjoy it. So make sure, I think it's going to be a big meeting. I booked about 700 rooms for 4 nights, but I think I'm thinking that I need to call them and renegotiate contract and add some more hotel rooms in the hotel. There are about 800 rooms, and I'm thinking that we might take all of them, because I think there will be a lot of you. We have a big international group going to come. Remember, it's a joint meeting with JPS, so there will be a lot of Japanese, but my gut feeling is that we might even get a lot more people from China, Europe. I've been talking with my European colleagues, and of course, we Americans will be there in big force. So I think there are 2 things for this meeting, coming next year's meeting, which starts on December 8th for 4 days, Monday, Tuesday, Wednesday, Thursday. We changed the sequence a little bit intentionally so that you can come. Obviously, you'll come on Sunday, if not Saturday, and then meeting starts Monday at 8 a.m. But then, rather than ending it on Saturday, we end it on Thursday, so that you have half day Thursday and Friday to enjoy. Saturday evening flights, most flights back to continent come in the evening, so you spend Friday, Saturday, enjoy Maui or other island, and then fly back, reach Sunday, and go to your work on Monday. So I think it's a good plan, and bring your family. Remember, we are a family. APA is a family meeting. All of us are part of our big extended family. So come enjoy the meeting next year. Meet your colleagues from all over the world. And the last thing which we want to make sure that our future of pancreas is not me. It's the young people. So you need to encourage your young people, your students, your postdocs, your fellows, your residents to come join, because that is our future. We need to train them. They need to know each other. They need to know senior people, junior people, and to encourage that. This year we gave 50 travel awards. Next year, between JPS and APA and IAP, we are going to give at least 100 travel awards for young people. So encourage your group, encourage young people. Even if you don't want to come, I hope that will not be the case, but send your young people to the meeting. So 100 of them will get travel awards. So enjoy the rest of the day. Thank you for joining, and look forward to seeing you somewhere, sometime, if not before, then in December 2024. Thank you.